Thank you so much for the invitation, and thanks very much for the introduction, James. Yeah, so it's my 10-year anniversary of being at the University of York now. So just for younger people in the audience, that is simultaneously amazing how far we seem to have come, and also terrifying how slow it seems to have gone all at the same time. So yeah. So just in case I run out of time, I thought the first thing I should do is make sure that I thank all of my wonderful research group. So yeah, they're all amazing. I'm not going to have time to talk about everybody's project. I'm trying to make it a coherent story, but they're all absolutely fantastic. I'm really lucky to have such good people working with me. So as with most of us here in this audience, I'm sure I've generally, my group and I are interested in biofuel redox enzymes because we think of them as like this holy grail of transition metal catalysis, and I'm sure I'm preaching to the converted, so I don't need to tell you too much. So if we think about these enzymes as the holy grail of biofuel catalysis, then two of my collaborators, who are my first kind of Indiana Joneses, so boldly exploring this field, Professor Paul Walton and Professor Gideon Davis, who are colleagues of mine here at York. And Paul and Gideon introduced me to lytic polysaccharide monooxygenases. So these are LPMOs. So these are monocopper containing enzymes, and they do this really important conversion of breaking the glycosidic bond in otherwise really highly recalcitrant carbohydrate substrates like cellulose. So these enzymes are now used in real industrial cocktails and they play a really important role really in doing the first step in industrial bioethanol production. So it's starting to open up like the really crystalline regions in cellulose, and then other enzymes can play a role. And there's a big mystery in the literature really. Historically, these were thought to be oxygen powered enzymes, absolutely classic redox metalloenzymes there, assuming copper's reacting with O2. But it's subsequently been shown that they react much more rapidly actually with hydrogen peroxide. So now there's a really big question about how these function exactly in nature. And it's important because if we understand the precise co-substrates and what tunes this reactivity, we can think about really optimizing this important industrial process. And so what my group has been doing with these enzymes is what we do with many enzymes, which is we get a purified metalloenzyme and we suck it up in a pipette and we splat it onto a carbon surface electrode. It's exceptionally high tech. Well, it's a little more high tech for studying the LPMOs. So we actually adsorb these down onto multi-wall carbon nanotubes. That does cause us some problems. This massive peak actually in these experiments is a background signal, but we can see alongside it smaller peaks which we can attribute to the reversible 1 electron electrochemistry of the LPMO enzymes. So we're seeing the electrons going in and out of the copper active site in these kinds of electrochemical experiments. And then what we can do is we can look at the reactivity of these copper active sites with hydrogen peroxide. So this isn't actually hydrogen peroxide and also a carbohydrate substrate. This is just looking at a non-physiological reaction that we can use to judge the catalytic competency and rate of turnover of these enzymes. So we can look at the copper essentially receiving electrons from the electrode and passing those on to hydrogen peroxide to turn that into water. And what I find fascinating is how we might be able to ultimately use these kinds of electrochemical experiments to really start to unpick structure and function. I've always absolutely adored all the structural biology work people do in the field of metalloenzymes. I collaborated with Juan as a PhD student, and I was just absolutely blown away by the beautiful structures they could get. And what I hope to do with electrochemistry is to provide some kind of complementary live visual data about how fast these things can react with electrons and exactly what kind of chemistry they might be doing. So what's interesting here is that out of nowhere, our AA17 does not want to do this hydrogen peroxide reaction in this way on our electrode at all. And there isn't really anything particularly obvious on the face of it about the copper active site in an AA17 that explains that. And then my second and probably original, I guess, love of transition metal catalysis, like most people here, is hydrogenases, which we're obviously all fascinated about. And as James said, it was the epic Fraser Armstrong who first introduced me to hydrogenases, so it's his fault I'm here. And again, one of the things I've always loved about hydrogenases is I've loved that we can stick them on electrodes, and you can actually in real time watch the electrons flowing through, and you can see the reactions with inhibitors, and you can see the catalytic bias. And again, I think what's really interesting to me for a long time is the fact that you can see an identical conserved active site in these enzymes, but see different substrate inhibition profiles and see different catalytic biases. And I've always thought that that's cool, because it shows how much the rest of the protein structure is really, really important. And it's not just about very simple organometallic chemistry. There's a lot more to the tuning of these enzymes. In particular, we've more recently been thinking about the active sites of hydrogenases in my group, because I've had Sol, who's just finished his PhD, working for me and my colleague Neil Hunt. And Sol's been using Neil's technique of 2D IR to look at the E. coli hydrogenase 1 active site. So this is a 2D IR spectrum that's been measured on E. coli hydrogenase 1 at the RAU central facility here in the UK. And there's a couple of key things that you can pull out from this. So first, because of the difference in the relaxation of the water bands versus the relaxation in the cyanide and the CO bands, doing this kind of pump probe technique lets us see far more detail in the cyanide region than it might be easy to see in a traditional FTIR experiment. And because we can see this lovely detail in the cyanide region, what we can do is we can make some unambiguous COCN assignments about the active site states of our nickel hydrogenase. So we can start to really pull things together about which carbon monoxide and cyanide bands belong to the same state. And because in these experiments, what you can see is you can see the decay in the ground state excitation, and then the kind of observance or the arising of these higher vibrational states. Looking at the distances between these states can give us information about the Morse potential. And then from all of this, what you can start to put together is these really beautiful complex energy level diagrams that can tell you about the vibrational relaxation pathways in these active sites of these enzymes, which physical chemists go absolutely mad for. We can also look at the pump probe as a relaxation technique, so we can look at the rate of decay and get relaxation dynamics. I think when you look at how the relaxation dynamics of an enzyme active site, so this is regulatory Ralstonia hydrogenase, which Sol worked on at the start of his PhD. If you look at that compared to active site mimics, which have been put into different solvents, you can see that clearly, just as the solvent plays a massive role in tuning the vibrational dynamics, by having that protein structure is achieving something really specific about the tuning of those vibrational dynamics. But what is irritating is that the different active site states of our E. coli Hyd1 all have really essentially identical relaxation dynamics, and also actually the E. coli Hyd1 is extremely similar to the Ralstonia. So again, there's really cool techniques we can use to look at the active sites. The active sites are fascinating, but they're definitely not the full story about how the catalysis of the whole enzyme is tuned. So as an electrochemist, what I've become increasingly interested in is thinking about the question about what role the electrical wiring of the protein will play in tuning the active site chemistry. And I mean, ultimately, I guess one of the holy grails for me is to see if our techniques can get good enough at at helping us unpick if there's a role of electron transfer in driving proton supply, because the electron proton ratio in most redox enzymes is really critical in controlling the products that get formed. And this is based on some work that goes, you know, my fascination with this is based on work that we started right when I started in York. So Lindsay was my first ever PhD student, and she did some work on HYDE1 in changing the secondary coordination sphere of the proximal iron sulfur cluster. And when Lindsay made these mutations, what she could do is we could use electrochemistry to look at the change in the hydrogen production of the enzyme. And I admit this is a subtle change. If you look at the native enzyme compared to the E73Q variant, we haven't made the E73Q variant suddenly a phenomenal hydrogen production catalyst, but we have definitely made it a better hydrogen production catalyst. I think that's pretty bonkers that a single amino acid change in such a big metalloenzyme can change the bias towards hydrogen production while conserving the oxygen tolerance. And it's suggesting that there is something in the way that we can think about the tuning of these iron sulfur clusters and play with the active site chemistry. And then my second PhD student, Hope Adamson, we moved a bit further along the relay and we looked at changing the secondary coordination sphere of the distal iron sulfur cluster. And again, we could see changes to the catalytic bias and the overpotential of the enzyme. So again, we were inspired to try making amino acid exchanges that were somewhat present when we subgrouped the enzymes into oxygen tolerant and oxygen sensitive enzymes. And so we could make an R193L variant. And when we looked at that variant, we could see it had better hydrogen production rates in our electrocatalytic cyclic voltammograms. And we could also see a decreased overpotential in our hydrogen oxidation in cyclic voltammograms. So we can see the power of sticking enzymes on the electrodes and looking at the catalysis, and we can get interesting equilibrium insights into the energetics of the perhaps putting electrons in, but we're really lacking kind of kinetic data. And historically, using electrochemistry, we can either look at catalytic signals in the presence of substrate, or we have to look at non-catalytic signals in the absence of substrate. And obviously what I'd really like to do is I'd like to look at the whole thing together. So we've been working now with Alan Bond for many, many years, who's an amazing electrochemist at the University of Monash in Australia. And Alan has really pioneered the idea of doing these slightly more complicated voltammetric experiments. So in traditional cyclic voltammetry, you apply a linear potential time sweep, and I always apply that to an adsorbed enzyme. But what we can do instead is apply a slightly more complicated potential time oscillation. And so this is the sum of an oscillating sine wave and our linear ramp. And we can collect the current that flows as a function of time in response to this input signal that we apply to our enzyme on our electrode. And then we can do what's called a Fourier transform to transform the current into the frequency domain. And then we can do band selection and inverse Fourier transform. And if we do band selection and inverse Fourier transform of the current that's flown as a result of a zero frequency input, we can get out all the same information that we would get out in a standard linear voltammetry sweep experiment. So we can do experiments in the presence of substrate and get out the catalytic electron transfer that we would normally see. But when we go to these higher harmonics, we call them, and we do a band selection and an inverse Fourier transform of these signals, we now see current that we can tune the experiment so that this current just reports on very, very fast electron transfer processes. So in a single experiment, if we can set it up correctly, what we can try and see is we can see the electrons that are trickling all the way through to the active site and look at the rate of chemical turnover in the active site. And we can also look, hopefully, at the electrons that are just hopping in and out of electron transfer centers along a wire in the enzyme. That's the theory. And most recently, we've applied our Fourier transform voltammetry technique really powerfully to looking at a very simple, well, on the face of it, a very simple disulfide redox reaction in a protein, actually from HIP-D, although its role as a hydrogenase biosynthesis protein wasn't the focus of what we've done here. So on a simple undergraduate level, you would describe disulfide bond breaking and formation as just a 2-electron 2-proton process, but as electrochemists we think about all the different substates that can exist, so we can think about how those electron proton transfer steps might be coupled. And the power of the Fourier transform voltammetry technique is that if we compare our experimentally measured signal, so that's the black line here, to different redox reaction models, we can work out which redox reaction model is more accurate. So we can, what we're comparing here is the blue line shows best fit when we've assumed that the one electron goes on and then the second electron goes on as two distinct single electron transfer processes, and compare that to the accuracy with which we can fit the data using the electrons moving simultaneously together. And so that tells us that we've got crossed potentials. It means that the thermodynamically, once the first electron has moved, the second one goes spontaneously. So it tells us about the relative values of the redox potentials associated with those steps. And what we can see is down here, we've got the residuals. So we've got the difference between the best fit data and our experimental data. And consistently at pH 9, across all of our harmonics, our 1 + 1 electron model is always a better fit. And we can repeat this across all the different pHs that we can interrogate the protein at, and we can end up developing these absolutely psychotic Horbet diagrams, which I was in charge of colouring in. So yeah, they basically look like a mad woman's being involved, because she was. But so what we can start to see is we can from our modelling data, we can extract reduction potential values and see how these change with pH. And that can let us then map out how the protein must move, how the redox reaction mechanism must change across the different pHs. So we can work out how we move across this diagram, and the reactions change as a function of pH. So that's obviously looking like we can start to unpick powerful stuff, and we can see how it's quite crazy when you think, I think when you think pH 4 to 9, as a biochemist, that's fine. You're just making a few different buffers. When you think as a chemist about the orders of magnitude in proton concentration changes that you've got across there, the fact that biology has evolved the surrounding active site of that protein to so tightly hold it to one electron across there, you start to see how the tuning of the proton electron transfer across biology can be very tightly regulated. So I think this can be a powerful methodology, but the limitation of it is I've got no reaction rates whatsoever for these electron transfer rates, because we can only analyze the 9 Hertz Fourier transform voltammetry data, and at that rate of oscillation the reactions are all at equilibrium, the electron transfer reactions, but we can't look at the higher frequency data, because modeling the higher frequency data, running the best fit parameter optimization takes just far too long to be practical. I mean, we could do it, but it would mean I'd be paying a PhD student to just lay in bed while a computer hummed for many days. So it doesn't seem efficient. So what we've been working on is trying to see if there's an optimum between how quickly we can analyze our experiments and how much information we can get out about the electron transfer processes. So at the high end, we can get loads of information out now from our Fourier transform voltammetry experiments, but their analysis is really slow. There's still loads you can get from classic direct current voltammetry experiments that you can extract very quickly, but you do have a problem that the background current of the electrode is very large, so sometimes you've got a lot more error on the Faradaic information you're extracting. So we've been trying to integrate into our toolkit what we're calling pure sinusoidal voltammetry. So now we're just looking at sinusoidal oscillations in voltage as a function of time, and the advantage is that because we're not doing that voltage ramp at the same time, computationally, because we've not got two time variables to consider, the time of the sinusoidal oscillation plus the voltage ramp, it makes the computational simulations much faster. You essentially don't have this catastrophic drop down in calculation speed when you start to go to high frequencies in your experiments. So we proved this worked by taking the electrochemist's favorite, ferrocene, and wiring onto the surface of our electrode and doing some modeling, and we could prove that the computational time to extract the best fit reaction model parameters decreased from 6 hours for our Fourier transform voltammetry to 30 minutes for our sinusoidal voltammetry. So there's a helpful added bonus of having this in our toolkit of experiments, and because it's much faster to simulate a sinusoidal voltammetry experiment, excitingly what that means you can start to do is you can use something called Bayesian statistical analysis to start to explore your parameter space. So these plots show us the confidence limits with which we can define different ones of our redox reaction parameters. And when you look at how different parameters interact with one another, you can start to see where you've really got parameter compensation based on these diagonal lines between two parameters. So ultimately, I hope this is a toolkit so that when I'm dealing with enzymes where we're not sure what the redox reaction is, we'll actually be able to visually compare the accuracy with which one redox reaction fits the data versus another redox reaction, and start to pull out where our uncertainty sits and where we might have problems with our data, basically where we need to do more experiments to have enough confidence that what we're saying is correct. So we're now trying to integrate this sinusoidal voltammetry into our protein electrochemistry toolkit. We've been working with an amazingly simple cytochrome that we're working on with our collaborators from the University of Leeds, and my PhD student Alice has been looking at some of the voltammetry of this, and Henry, my postdoc, has now been able to work out an optimal analysis kind of like flow chart to combine different information from different voltammetry experiments to be able to get to a really good fit to the data in as fast a time as possible, so that we can now actually extract kinetic parameters for the rates of electron transfer in a relatively time efficient manner. And of course, my big dream is that eventually we have a way of doing an experiment in the lab, having a go at simulating it, and then working out how we should improve our experimental parameters to improve the confidence with which we're measuring things. So work out how to tune in the frequency and things of the experiments. Currently, Henry's starting to ask how much of an advantage adding in impedance spectroscopy and how much that can start to help as well. And we've just started collaborating with two guys from Bar Ilan University in Israel, and along with Alistair, they're now using, we've started to work out the advantages of using sine wave amplitude to tune in and out of seeing the thermodynamic dispersion of our enzymes and our electrodes, which I'm really excited about, because I've got a feeling that the enzymes molecules can probably talk to one another. Particularly, we know hydrogenases really prefer to be in multimeric structures, and it's going to be really interesting to see how much we might be able to ask questions about that. So that's kind of stuff we're doing in my group focused on our technique development. I've obviously got loads of lovely collaborators, and I've got loads of lovely other students whose work I've not been able to talk about today. So my group is part of our chemical biology group in York. So there's myself and Martin Fascioni and Leanna Willems and Chris Spicer, and all of our groups work together. I've got some PhD projects that are available at the moment, and there'll be postdoc positions being advertised as well. So if you like what you see and you'd like to know any more, please do be in touch with me. That'd be fantastic. So yeah, thanks for your attention.